Okay, hello. Thank you for the introduction and the invitation for today's workshop. And today's talk will be titled Antibody Fragments, Engineering and Possibilities. I first would like to acknowledge my lab. We are not many people, but we work hard. Some of those students graduated already and they are abroad. Most of the work is done by Merve Arslan that I will show you today. And in our lab, we mostly work with SCFV, single-chain variable fragments, and also we have a project on nanobodies. But today, I will talk about one of our developed SCFVs and specificity engineering. You know, the specificity is very challenging in antibody world. So we try to engineer that. People usually engineer affinities, stabilities, other things, but specificity is always a challenge. So you already know what an antibody is, but I try to schematize it here. It is a very big molecule, as you know, 150 kilodalton. But when you look at the SCFV part, we call this VHVL with a flexible linker, SCFV part. It is very small, 30 kilodalton, and in here it is in color. And it is small, and it is easier to engineer. And also, after you engineer this part, you can obviously make it bigger as IgG after your engineering efforts. So it is easier to convert. So we already talked about antibody fragments, and the most famous one is a nanobody over here, and SCFVs that we work on. And nanobodies are even smaller, that's good. Smaller is always better in protein world. Not always, but mostly, because it can, in therapeutic approaches, it can penetrate the tissue better because it is smaller. And there is no post-translational modification N-glycan site in the full length. You know, there's an N-glycan site at position 298. And there is nothing N-glycosylation here. N-glycosylation is always a problem in mammalians expression. Mammalian production, because sometimes the glycans change upon different colonity, and you need to deal with that. And before you go into therapeutic approach, you need to make sure every production is the same. But without, that stochastic part, you are only the amino acid sequence. Always you will have some oxidations and other things in your protein, but it is not as bad as the glycosylation. So also, as already we talked about, it is easy to produce. You can use a microbial cell, which is way cheaper. If you compare 1 liter of mammalian through cell line, media is about €100, and €1 or €2 for the E. coli or the yeast, media. So it is 10 or 100-fold difference in the production cost. So it is easier and cheaper. And as I said before, after you engineer this part, you can definitely convert it to IgG or other parts or other engineered formats. So right now, I just checked the website yesterday. There are 191, more than 190 antibodies already approved on the market to use in different diseases, mostly cancer. Half of them are cancer. This is the red part. And the other part is mostly autoimmune diseases and some eye diseases. There are some other less percentage of other diseases, but mostly cancer and autoimmune diseases. So as you can see, this is exponential rise in the therapeutic area. Small molecules are not good, as you know. For cancer, I can say chemotherapy, radiotherapy, yes, they are efficient, but they have lots of side effects. And right now, we are in the area of biological drugs like antibodies here and some others. And antibodies are very successful at targeted therapy. So there are lots of, in drug companies, they use lots of antibody technologies. And also in Turkey, my main aim to return to Turkey is to actually make this happen. So hopefully, before I retire, I will make an antibody on the market originally developed in Turkey. This is my main aim, actually, in Turkey, because there is no original drug developed in Turkey until now, even small molecules, always degenerate. So yeah, we can do that. I believe that. And among those 190, 12 of them are antibody fragments. Most of them, I think 9 of them are Fab fragments. You know, the Fab over there. This fragment in the market. 2 of them are SCFs and 1 of them are nanobodies. As I remember, I can be, you know, like wrong, but estimate numbers. And there is 2 diabodies, as I remember. Diabodies are the like SCFs, but 2 of them. And the nanobody approved is also like a diabody, 2 nanobodies, not 1. Okay, so this is just a general scheme for antibody discovery. In our IBG, İzmir Biomedicine and Genome Center, we both do hybridoma, and also we just started a project, single B-cell cloning. We are very excited about that. We just immunized the rabbits like a month ago, and we will try to get some antibodies from those immunized rabbits. Single B-cell cloning, I will not talk about it in my rest of the talk, but it is very exciting because most of the drug companies use single B-cell cloning for antibody discovery because it is more efficient, more high throughput, and you know, it is way better than the hybridoma technology. But in the research lab that you use, antibodies, different lot numbers, everything, they come from mostly from hybridoma and supernatant of the hybridoma. So from lot to lot, there is a difference. So sometimes it will work in your Western blot, and you buy a different lot next month or 2 months later, it will not work, or it will not work as good before, because it is not regulated. It is just directly from the supernatant of an hybridoma, and they do not control. They just do protein A purification, and they just sell it. So that's why now there is a call saying that Plinkton, maybe you know him, he's like the father of the antibodies. And all antibodies should be recombinant. Even research antibodies, not only therapeutic, should be recombinant, obviously, because you need to know the sequence and everything. But also research antibodies should be recombinant. Otherwise, it cannot be regulated, and it will not be reproducible, your results and everything, including diagnostics. So as you can see, I like this image a lot. And this is the antibody iceberg. As you can see, there's a big part under the sea. And those parts, mostly nanobodies, that we are here together discussing nanobodies. And also, you know, display techniques, mostly phage display and other display techniques like surface display that we do. And they are under the iceberg right now because they are not heavily used in the research area, but they are coming through, hopefully. And there are some other scaffolds, like antibody fragments. And there are also some deep sea of lost opportunities because of the opportunities of those things. So today we use polyclonal, monoclonal antibodies, but they are not, you know, like useful. We need to go in deeper and, you know, unravel this part of the iceberg for sure. Sorry. Okay. When I say recombinant, it means that you know the sequence of your protein. Otherwise, it will not be recombinant production. So if the sequence is known, protein engineering is limitless. You can do anything with it. You can just modify the sequence. You can make it shorter. You can, you know, left out a domain, and everything is possible as protein engineering. And I call it antibody engineering because I mostly work on antibodies. And as I said, we mostly work on SCFs, and here is the schematic of the SCFP here. There are 3 CDRs, 4 framework regions, but I think there is also a CDR4. I will talk about it later. So those framework regions are important, yes, and there are some very important, you know, stem of the CDRs that we call Wernier zone residues. Okay, in 2018, Nobel Prize was given to you know, people who work with phage display and using them for enzyme engineering and also the antibody engineering. So those 2 professors are for the antibody engineering and this professor for the enzyme engineering. And it's actually opened, the display technologies opened a new era for the protein engineering world. Yes, it is for sure. And it is limitless. You can do anything. That's the opportunity, actually, for therapeutic, for diagnostic, for research area. So the only thing that you need to do is know a little bit structure function of the antibodies. If you change the amino acid sequence, you change the structure and you can change the function. So there is a, there should be a fine tune between those two. When you change something and you expect something, you need to be very careful. You change everything, structure, function. So you need to screen for everything. And you need to select where to engineer according to structure. People usually focus on CDR regions, but no, you don't need to engineer only CDR regions to make a specificity of affinity change. You can also change the framework regions to make those changes. Okay, so in this talk, I will just try to give an introduction of our research where we try to develop a dual specific. So by using a region-specific antibody engineering approaches. So what is dual specific? You probably heard of bispecifics. Okay, this is bispecific antibody. This is a diabody, which can also be bispecific by changing this SCFP. But dual is something else. With one domain, you can bind two different antigens. So it is a bit surprising, how can it bind to the two different antigens with only one set of CDRs? It is, a bit hard engineering question, but we can do that because there are some available paratop regions on SCFEs or on IgGs, variable regions, which can also bind to another antigen. There can be some steric clashes. It cannot bind two at the same time, but it can bind at different times. Even if they are small enough and there is no steric hindrance, you can bind two at a time. Yes?
You are not talking about binding to a complex, protein complex, right? You are not talking about binding to...
No, not complex, just the monomer. Yeah, monomer, yeah. So for cancer, there are lots of targets, as you already know, and that I showed you before. And there are 190 antibodies, and half of them are cancer-specific. There are lots of biomarkers for cancer. And we happen to develop an anti-VEGF SCFP, which is already used in cancer treatment. Maybe you already know the name bevacizumab, which is very popular anti-cancer drug, which is usually used in combination with chemotherapy, but it is effective in most of the cancers approved on the approved for, And we already have this. What is VEGF, vascular endothelial growth factor? Basically, it is a soluble factor, and which can be secreted from the tumors. But during development, it's already is secreted because it does vasculogenesis. It makes new blood vessels. When you secrete, the cells, epithelial cells secrete VEGF, it makes that, okay, more epithelial cells of the new blood vessels are forming because there is a need. So maybe you already know this, but the first cancer doctor noticed that when they opened the patient and see the tumor, there were lots of blood vessels around the tumor. And it was the first reporting of that, vasculogenesis around the tumor. Because the tumor needs oxygen, tumor needs nutrients, right? And how to get that? By getting more blood vessels. So tumors are evolved to express VEGF around to make the blood vessels come to their places to get more oxygen, to more get nutrients. So in women, we already, do vasculogenesis during our periods, but like in the normal human body, we don't secrete much VEGF because we don't need new blood vessels unless we are injured or something. Okay, so another target, because we want to develop a dual specific, we chose PD-L1 because also we have another 1004 project, which started in the same time with the Istanbul University. And we also tried to develop an anti-PD-L1 antibody for cancer treatment. That's why actually we chose PD-L1, but not the only reason, but because it is efficient in cancer treatment. Basically, it is, I will just show here. There are lots of anti-PD-L1 antibodies in the clinic, and also there are some coming through in the clinical stages or preclinical stages. As you can see, most of them are bispecific. So PDL works as monospecific, and some patients' survival rates increases with anti-PD-1 or anti-PD-L1 treatment. But even if you make 2 targets, it is even better, right? Because you hit one bird with, one stone with 2 birds. Okay, that's important, and they are coming not only for the PDL, but most of them, they try to do bispecific. Okay, so what is Werner zone residues? So it is like very underestimated in the antibody literature, unfortunately. And I try to resurrect Werner zone residues for the antibodies. Why? Basically, this is just a 2D schematic of an, VH and VL regions. And basically, those cyan regions are Werner zone residues, which were stated back like 1988 as called Werner zone residues. But nobody updated that. They just stated that. And we just saw a couple of papers which says important for humanization. What is humanization? We already talked about this a little bit in the previous talk, but basically we mostly discover our antibodies from a mouse or rabbits, right? And when you get that, you cannot just inject it into human because it can be immunogenic because it is different sequence, different structure. So basically, you need to humanize your sequence, mostly framework regions and other FC regions, to make it less immunogenic to human. And humanization is a bit like stochastic approach. I try to do that, but my computational background is not that strong, so that I cannot do, so I need help. Actually, for our 1004 project, we used a service from China for that purpose. But we can do that. We need to just, with some computational collaboration, we can do that, I think, because when we check the Werner zone residues, those are the residues, most of the back mutations happen. What is back mutation? For humanization, basically what they do, they get the CDR regions in the red here, which is the antigen binding part. They just copy and paste that thing, that CDRs onto human framework, okay? But it will not work because loops and framework regions should work together to make it stable and binding to their target antigen. When you copy and paste that, it will not work. So they first do that, and they see that, okay, affinity lost. Then they said, okay, change this stem, change this framework region and see what happens. They change it to, if it is from mouse, back to mouse sequence to make it like more, protect its mouse affinity. It didn't work, okay, change the other one, blah, blah, and it is like very stochastic. And most of those mutations are Werner zone residues. So, and if you search the literature about humanization, you will not see much because it is mostly the, like biotechnological companies who hold those as secret. Their algorithms are very secret because if they give those secrets, humanization secrets, everybody can do that, and it is not that open. They are not that open. So basically, when we check the Werner zone residues, they are the point of back mutations. When they do back mutations, at some point they will get the affinity of the original molecule, original antibody. Okay, this is enough. Or they will just do more engineering or something like that to get that affinity. So meaning that Werner zone residues are very important for affinity, for antigen affinity, and also specificity, I think. And so basically, as I said, mostly neglected, but it can affect the conformation of the CDRs, which will affect the antigen binding. So in our study, we engineer a part of the Werner zone residues And we try to develop a dual-specific SCFE. So what we did. So this is a bit complicated slide, but I will try to explain it to you. So basically, this is our SCFE with VH and VL part. And this is our anti-VGF SCFE, by the way, that we already developed. We try to convert this to dual-specific, which will bind to PDL. So first, we need to choose a region where we will engineer and create our library, right? So to make PDL bind without losing the VGF affinity. So when we check the 3D structure, we see that VGF binds mostly on the heavy chain CDR3 and heavy chain other regions. But the light chain part is mostly empty. So when we zoom in here, which is we called a light Werner zone 4, it is mostly in interaction with the light chain CDR1. It is not interacting with the VGF. Also, light chain CDR1 is not interacting with the VGF. This part is completely empty. Okay? So if we focus on this empty part where VGF doesn't touch, can we make PDL bind to this here after we engineer? Okay? So basically, also, I put CDR4 here. This is a bit provocative, I know. There is no CDR4. But it can be a CDR4. Why? Because it is the only loop which faces to the antigen site. Okay? So CDR1, CDR2, CDR3, they are all facing towards antigen. And this is the only loop. We call it framework 2, by the way. But I call it Werner zone 4 because of the lots of Werner zone residues here. And which can be engineered to make it like a CDR. Maybe in evolution, it didn't get hyper variable in evolution, but we can do it because we can engineer it. And also in nanobodies, I think the same thing happens. They have another loop right close to their CDR1, which is also called framework, but it can be engineered because it is a loop. Loops are flexible. So if it is flexible, it can tolerate some mutations. So that's why we focus on this part, and we try to engineer that part. So our approach is using a surface display. Basically, we mostly use PICA Pasteurist system. And in PICA Pasteurist, AGA1 in Cerevisiae is homologous to SEG1 in the PICA Pasteurist. And we have our SCFE with the FLECTEC at top. And we check the antibody expression first, then the antigen binding, because the starting SCFE is the anti-VGF, and it mostly binds to VGF. And then we try to get the KD of our anti-VGF. Actually, we already know the KD of our SCFE, affinity of our SCFE to VGF with SPR technique. But we also did it with the flow cytometry. And it was very close, but a little bit off, but it is like in the same range. It was 5, I will show in the later set, but 5 nanomolar in the flow cytometry in this experiment. But it was in SPR 2 nanomolar. It is in the same range. So after we confirm that our SCFE on the yeast surface has the affinity for VGF, we create our library on that loop that I showed you. And we mostly focused on the 68 and 69. This is only 2 positions I know, but we did other libraries, but it is in progress. We only checked the first library. And after 4 different rounds of sorting, we got a cell population. And after we sequence those, we show that there is actually some enrichment for some of the amino acid combinations. And also, we checked the PDL binding for those, and we increased the PDL concentration to see if it is really binding to PDL. Yes, it is really binding to PDL, those mutants. So it was very exciting, those results. But also, we need to check how good it binds. Is it specific or not? And also, we checked the VGF binding after mutations, and it also binds to VGF. It didn't lose the VGF binding. So I will get some water. If you have any questions, you can ask me, right?
Can you describe the PKA system, how the expressionists have
Yeah, it is methanol induced. So yeah, it is induced.
Sorry, you are screening against PD-L1 in the presence of VEGF ligand, right?
No, actually,
Why?
because we already know that it binds to VEGF.
But if the binding of PD-L1,
If there's a competition,
Yes, but as start of the talk, as I said, it can be, if there is no steric hindrance, it can bind together. But if there is a steric hindrance, it can bind one or the other one. It depends on the paratop hindrance, right? So our approach was to, prove our proof of concept. It can bind. Right now, we are also trying to do some competition assays, and it seems like it is competitive binding. So yeah, it cannot bind two at the same time.
so it's not important. You just
It is not important. We just try to see if we can engineer an available surface of an antibody to bind to a second antigen. In B specifics, because there should be two separate domains for double specificity. In our case, one unit, two specificity. Yeah.
So are you planning to use another screen in the presence of VEGF also? Do you think you will get to the...
But in that case, probably because it binds to VEGF better, because it is originally developed for VEGF, it will just kick out a new antigen and bind to VEGF, and we will get the same thing as VEGF binders.
But I think there is also still available region in the CDR4
Yes.
is also available, although it binds to the VEGF.
Oh, you see two VEGF at the same time? No.
The VEGF is one, but
Uh-huh.
the CDR4 is available for binding PD-L1. So in theory,
In theory, it can happen, yes. It is a good suggestion. Yeah, in theory, yes. But we also check the steric hindrance and computationally, and they clash a little bit. That's why I was like, okay. But for smaller antigens, yes, you can do that.
there was an antibody series in Portugal online conference. So there was a guy from Chugai Pharmaceutical. So they were screened for against a ligand that binds to this ligand in the presence of ATP. So they just select for a clone that recognize ATP and then screen for another ligand.
Yes, it is a small molecule that, yeah, it can happen. It's for small molecules, yes, this proof of concept can work, and even better because it can bind at the same time. Yeah, thank you. So also we checked to see those mutants that we got, most present mutants, if they lose VEGF binding, and they are in the same range. There is not significant difference between them. For the PDL binding, only NQ was better than the wild type, the NQ. Actually, it is not that enriched here, NQ, but it was way better than the other mutants that we got from the library hit. And here are the KD values, and also we used flow cytometry for those. As I said before, for the wild type, we were around 5 nanomolar KD, and for the NQ mutant, it is in the same range. But with SPR for the wild type, it was around 2 nanomolars, 1.8 as I remember. And so they are in the same range, and we didn't lose VEGF affinity at all. For PDL binding, so this was the exciting part. And the affinity was not in the therapeutic range, obviously, but it is still good. 51 nanomolar, it is not bad. We can do some more engineering to measure the affinity, because we only check the 2 positions, and we plan to do more than 4 positions in the same loop. So this can be increased. And as you know, for the wild type, it doesn't bind to PDL at all, non-specifically. Specificity is high for that VEGF, we already know that, but we also checked as a control. And there is no binding for wild type, but for NQ mutant, it is 51 nanomolar. So that's good. This is the proof of concept, as I said. It worked, so that loop is open to engineering. And there is not only that loop in the light chain, also there is another loop in the heavy chain in the same position in the framework too. Okay, so as I said, engineering of antibodies and antibody fragments are limitless if you know the sequence, okay? So display technology is the best way for now for those type of high throughput engineering strategies, but also computational strategies can, improve and help a lot. Actually, we use computational tools a lot. So without computational tools, most of them we cannot do that. And as I said, our antibody is not in the therapeutic range for the PDL binding. It should be at least in the range less than 10 nanomolar KD for the therapeutic range. And also, as I said, B specifics are coming. Why not dual specifics? So why this is important? B specifics is like, even if they are in the same protein domain, two different variable regions, it means, right? So production also can be affected from that, right? It seems like it is like producing two proteins in the same pot, okay? And there should be lots of troubleshooting for that. Even for monospecific IgGs, it is hard to produce them. But for the B specifics, it is even harder because folding of one region can be slower than the folding of the other region. So the cell lines cannot cope with that, and the production yield might be lower than the monospecifics. So by using dual specifics, you only deal with one sequence. So that's the advantage. The folding is the same. So and the production yield would be the same, and everything, all the regulations would be the same, and in vitro characterizations. As I said, we chose a Werner zone loop. We call it Werner zone loop, but this is the first term that we used in the literature. It is not Werner zone loop. People usually call it framework 2, framework 1, and those kind of things, but we call it Werner zone region. And we showed that loop is available for the second antigen binding. And we have a lot to do, actually, but my student actually graduated and went to Belgium for her postdoc. Right now, the project stopped a little bit, but we will just continue, hopefully. And as I said, our proof of concept for dual specific worked. And thank you very much. Before, actually, because IL-6. We also developed an anti-IL-6 SCFP, by the way, for if you need a collaboration. We already have the sequence, and the affinity is in the picomolar range, by the way. Just keep that in mind. We already have it. And Mehmet İnan, maybe you already know him, the APIOC of the R1004. We are in collaboration with him, with Akdeniz University, to develop that. We already have the sequence, and our production yield is good. And affinity, obviously, is very high. So if you need it for your 1004, it could be a entire 1004 collaboration. Thank you.
It is really very interesting area in terms of opening a new avenue for the binding of antibodies. Maybe I can start with one question.
Yeah.
Now, I'm not an oncologist, but I'm a practicing rheumatologist, mainly dealing with inflammatory conditions and using these monoclonal antibodies very frequently. When you use an antibody, or when you want to use in combination, when you have two targets, they usually work together in terms of pathogenesis, so you want to hit both of them at the same time. In regard to PD-1 or PD-L1 and anti-VEGF antibodies, well, usually the anti-VEGF antibodies are used in a certain type of cancers in which we usually don't consider anti-PD-1 or anti-PD-L1 antibodies. So they are a bit different types of antibodies that are used for different kinds of diseases. So if you want to use anti-VEGF, you want to use the best one. But if I want to use also anti-PD-1 or PD-L1, I want to use something with much higher affinity, another one.
Yes.
So it may not be an interesting idea to me as a physician, having an antibody which may have also some affinity for anti-PD-1 or PD-L1.
So
maybe
basically,
it is
yeah.
a proof of concept, targeting something is really very nice. But in terms of the using these antibodies in practice, it is not so reasonable.
I see. But the thing is that the affinity is not always, higher affinity is not always better, by the way. You know, high affinity, yes, in the therapeutic range, it should be in the therapeutic range. But there are some studies showing higher affinity antibodies will not give the same in vivo efficacy. So because it binds so tight, it will not like, you know, like...
Well, it depends on how
yeah, it
you
depends
measure
on,
affinity as well.
yeah.
That's another issue.
And the thing is that actually, just last year, I think, Genentech, it was, they tried to develop B-specific VEGF PD-L1 antibody. Just they are developing, but we don't know if it has efficacy. This was after we chose the PD-L1. We chose PD-L1 because of the ease, because we already have the antigen, because of the 1004. It is easier to screen because we have everything under our hands, yes. But they did it, and they published already that paper. It was a bit like disappointing for me because I was not I didn't publish this work yet, and yeah, that's the thing. But yeah, you are right. It may not work or it may not work, but definitely bispecifics, or I can say dual specifics, are coming. When you check the preclinic, they are not doing monospecific anymore. They are trying to do different combinations because for example, for the bevacizumab, it cannot be used alone. It should be used in combination with other things like.
It should be used in combination with some chemotherapy.
Yes. But mono, one target is not enough. We already saw that, and people are trying to switch to different targets at the same time.
Sure, especially for anti-VEGF, it is not directly for cancer. It's an indirect way of dealing with cancer. But it may be a very good idea if you want to use this antibody to bind two different aspects of the same protein. I mean, if you are targeting
different isoforms, maybe, yeah.
Different parts of different epitopes of a protein can be targeted with this kind of dual antibodies. That would be a wonderful thing, actually. Much higher affinity, maybe, let's say, a better or long-term inhibition of your target can be achieved with this kind of thing.
Yes.
Instead of using two different targets, maybe two different epitopes of the same target, maybe.
Yeah. For example, PD-L1, there is also PD-L2, which is not highly expressed as PD-L1, but it is also expressed. It is also immune checkpoint inhibitor. We can use as immune checkpoint inhibitor. Why not PD-L1 plus PD-L2? It may work.
Or ligand and receptors.
Yeah. PD-1, PD-L1. Yeah.
Thank you.
Yes.
Maybe I use one problem that
Is it working?
one problem that I think is underestimated when using bispecific or any kind of multivalent system is that is not consider the real distance between the two epitopes. No? So maybe you have the theoretical capacity of binding to sites, but maybe in the reality, because of the geometry of the molecule that you use, you're not able. So maybe you can have an ability, especially if you have a very high density, like on the chip or the SPR system. But in the reality, maybe it's always working as a monospecific. Did you think about this?
Yeah. You are right. It can happen. Like it can be on the monospecific due to maybe, as you said, like the geometry, steric hindrance. It may not bind to the second antigen. But the thing is that, as I said in the previous question, actually we didn't expect to bind at the same time. So that's not a problem actually. So it can, for example, in theory, and our antibody is around the tumor microenvironment. And there's VEGF coming out from the epithelial cells because it needs blood vessels. because the concentration of that VEGF around that tumor microenvironment is very high, it will only maybe bind to VEGF. But at the same time, there is a T cell coming through, checking the tumor microenvironment. It has the PD-1, and the tumor cell has the PD-L1 overexpressed. When they interact, okay, it will say that, okay, it is good here. You don't need to attack here to T cell, the tumor cell. But because that it needs to overexpress PD-L1 to say to that T cell, right? So because that PD-L1 is also over there, overexpressing, our antibody will also bind to maybe PD-L1, not VEGF anymore, because it's overexpressed, PD-L1 overexpressed. It will go bind to that, and it will inhibit that interaction. And the T cell, oh, okay, this is something going off, I will just attack this. So saying that it doesn't need to be two at the same time. It depends on the concentration of our target in the tumor microenvironment. Basically, the idea is that. Yeah, but in vivo, we didn't try this in vivo. We don't know the efficacy, yes. But in theory, it doesn't need to be, two at the same time.
In these experiments, did you use the whole antibody or only
Only SCFP.
only SCFP?
Yes. But we can convert it to...
Just to extend Ario's question, what happens if you use the whole antibody? If it dimerises with heavy chain and light chain, what is the position of Werner zone loop?
The same, actually, very, very similar. Yeah, because there is only one flexible linker in the SCFPs, VHVL, and the same thing for the antibody. Actually, I will try to show. Ah, sorry, it was bad. So it is the same actually. This is the IgG VHVL, and this is the SCFP VHVL. It is the same, probably the same VHVL interface, but because to make it single chain, we use a linker to make that, covalent bond. But the orientation is mostly the same.
Well, it's not the orientation. The orientation of the Werner zone.
Oh, I see.
CDR 1, 2, 3, if the orientation is somewhere here, it may work as a single molecule, but when you dimerize it, it may be hidden somewhere.
Yeah, no, actually it is all the way up here. I can show like on the edge of the VL and on the edge of the VH, those Werner zone loops. There is no way that loop will just go inside the, it is not impossible at all. But as I said, it is not the only Werner zone region that we focus on that region, but there are
On the
other,
loop,
yeah, on the loop, yeah.
Any questions? Yes, please.
Yes.
Have any structural data saying that your heavy chain CDRs are binding to one of your antigen and the light chain CDRs are binding to the
No,
other
we don't
one?
have structural data for that. It's just the KDs and those kind of data. We hope that we get something, we do something like that in Turkey. The structural part is very, already know. Sorry, yeah, actually I was a structural biologist during my PhD. I did X-ray crystallography, but you need a lot of infrastructure for that.
So for our young investigators, there is an unmet need here. So there is an opportunity.
So when you talk about the bispecific, since you know about the industrial part, what are the tricks to prevent the monospecific in the cell line that's expressing it? Like where are the mutations that prevent the monospecifics from forming?
Monospecifics from forming what?
Like when you make a CHO cell line that expresses two VHs and two VLs to make the bispecific,
Yes.
you can still make the monospecific antibody, right?
Yes, monospecific is more straightforward than the bispecifics,
So in the
yes.
same cell line, right?
In the same cell line, yes.
So how you have to somehow exclude the monospecifics and purify the bispecifics.
How do you guarantee that you are producing bispecific?
Oh, I see, I see. So actually the problem, like for production, is that, mostly for monospecific I can say, sometimes CHO cell lines produce the heavy chain more than the light chain, or the vice versa, it can happen, but mostly that way. So that's heavy chain, there will be actually, you know, like, I will just show here. Where was my slide? Here. So basically, what they do is that they produce the same. Actually, they use two different vector combinations. As I have never done bispecific production, but from the papers that I read, they use two different vector system, dual vector system. One with one heavy chain and the other light chain. The other one with the one heavy chain, the other light chain. And they checked, after they purified that, they checked binding to both of the antigens to make sure only domain number 1 and domain number 2 is present in this IgG conformation, right? So if this is only domain number 1 and domain number 1 here, it will not go into their production line at all. So that's why the production yield is very low compared to monospecifics. So that I was trying to say. So you are dealing with two different things in the same cell line.
So all light chains are for one antigen and heavy
Yes.
chains are for another one.
But because you use two different vector systems.
Well, I think there is no more question. Thank you very much, Sibel.
Thank you.